You can start.
Thank you. Thank you for the very nice introduction and also for the invitation to speak. So today I thought I would tell you about purely theoretical work, and so this is work that was done with a limited number of collaborators. Usually I have a very long list of collaborators, but here this was work done with Daniele Conti, who was a postdoc with me at the NS, and with Ilya Nemenman, who's at Emory University. And so, okay, I have to stop sharing. This happened to me before. Sorry about that. So, okay, so the topic of the talk is about prediction in biology. So, as you know, it's a famous quote that predictions are difficult to make, especially when they're about the future. And so, I mean, this is a simple idea, but like you can only make prediction if the thing that you're trying to predict is correlated with itself in time. So in other words, if it's predictable, right? So to make prediction, you sort of need a model for how so you make a prediction about the environment as a biological system. And you need some sort of a mental model for how this environment will change over time, right? So which will be given by some evolution operator A, right? And many biological systems do this. I mean, one of the first thing that comes to mind is that we make predictions in our brain all the time. So for instance, if you think about Serena Williams trying to hit this ball, in fact, when she sees the ball, there's a bit of a lag between the light coming to the retina and then the information going to the brain, which is about 100 milliseconds. So by the time she sees the ball is already like 50 centimeters further. So she has to make a prediction for where the ball is going to be, right? So that's a very straightforward example. But there are many other examples in biology. You can also think of evolution as a prediction mechanism. So why is it a prediction mechanism? Because when there's adaptation and evolution, biological organisms basically learned from the environment what's going on, and then they adapted their behavior and their response, the genome, to behave in an optimal way, I mean, in a good way, in an adaptive way, later in the future. You can also view this at the level of phenotypic response. So if you think of signaling, of chemical sensing, or in chemotaxis, for instance, you learn about the environment by sensing the environment, and then you will tune your behavior and your reaction as a function of what you've learned, right? And finally, there's an example I'm not going to talk about at all today, which is the immune system. You can also view immune memory as a way to learn from past encounters with the environment, in that case, infections, and then adapt your immune response and your immune memory, your immune repertoire, to best protect yourself against future infections. So this is about prediction, and so what's the link exactly with adaptation, which is another thing of another topic of my title. So one way to view adaptation is to think about how to basically adapt your behavior based on what prediction you make about what the environment is going to be, right? So formally, let's imagine that there's a sequence of the state of the world, so that's what I call E of t, right? Going into the past, right? And so, as I said, then there's some evolution operator with which this environment evolves that will tell you what you can expect it to be in the future, right? E plus delta t. Now, the problem is that any biological system never actually sees the environment exactly. What it sees is some noisy version of it, or it sees something that's actually emitted by the environment that depends on the environment, but it's only observations that do depend on the environment, right? So you can view this as a degraded version of the environment information, right? And so then the biological system can try to use these observations to make a prediction about what it thinks the environment is going to be in the future, right? So that's the prediction part. And one way to do this is to use, for instance, Bayesian filters. And once one has this model, this prediction for the future, then one adapts the behavior by having an adaptive response whose goal is actually to maximize some objective, or minimize some cost, so maximize some reward. That's a function of what the environment is actually going to be, and what the response is, right? So this is a way of thinking about adaptation, is that you use the information from the past, you make a prediction about the future from that, and then you adapt your behavior to basically behave in the best way in the face of that future environment. So this defines some sort of an optimal strategy. So the optimal strategy is to actually use a Bayesian prediction and then to just maximize this objective function, right, as a function of time. So that defines, that gives you a mathematically well-defined problem of optimality. And I'm going to show two examples of this in the rest of the talk. Usually when you talk about optimality, that there's a natural question, which is, is it true that biological systems actually approach the best strategy? Just as a disclaimer, before I go any further, in general, no. I mean, biological systems are not necessarily optimal, even though, from the argument of evolution, you could argue that they try to reach a solution that's as good as possible. However, thinking about these problems in terms of optimality is useful to uncover design principles for how biological systems function. It helps you rationalize some of the behaviors that you see in biological systems. So it's a guide more than a hypothesis, right? So, so what does adaptation look like in real biological systems? So this was the theory, and this is more practical. So this is what you actually see in experiments. So imagine, one typical example of adaptation is bacterial chemotaxis. So bacteria such as E. coli manage to swim towards the chemoattractants, so food or things that they like. And it's known that that to do this, they can regulate the behavior of the motor as a function of the history of concentrations that they saw. Right? And so already back in the 80s, people did simple experiments where they recorded the behavior of the rotary motor of these E. coli, right? So basically what they recorded is probability that it spins in one direction versus the other. So that's the response, right? And they subjected these cells to a step function in the concentration of the chemoattractant, right? So this is what you see here, is a small step function at times 5 seconds. And what they saw is that in the beginning, you see a change of behavior. So you see, the probability of going in one direction goes up, but then after a while, it goes back down to some baseline level, right? And this is typical adaptation. And that's because by the time, by 10 seconds, the bacterium has now gotten used to the new concentration. And so there's no need to respond anymore, because what you can argue is that E. coli actually only cares about changes in the concentration, and therefore, if the concentration stays the same, then it shouldn't react anymore. Right? The second example, which is a very different one, it's actually drawn from neuroscience. It's the example of visual adaptation, but the phenomenon is very similar, right? So it's in fact the phenomenon by which, if you enter a very dark room after having been in the sun, in the beginning, you won't see anything. And that's because your visual system is not well adapted. And you can actually measure this at the level of the output of the retina. So by measuring from retinal ganglion cells. And what you can do is do a very similar experiment to what's done for E. coli. You suddenly increase the mean intensity. And then right after this increase, you see a huge response, right? But after a while, the system adapts, and the system gets used to this luminosity, and the response goes back to some baseline, right? And after that, if you could put them back in the dark, this is what happens to you when you enter a dark room, then it goes back down, and then again goes up to a baseline. Right? So these are two different types of adaptation, and what I want to try to show is how we can maybe explain this kind of traces and this kind of dynamics in terms of a model of optimal behavior and optimal adaptation with optimal prediction. Right? So let me start with this example of sensory adaptation. So it's motivated by neuroscience, but you can also think of E. coli as a possible example in which to think about this problem. And so this was this is work that we recently put in the archive a couple of weeks ago. So let me show you it's a slightly different version of this adaptation to the mean light level. And these are experiments done in the fly visual system as opposed to vertebrate visual system. And what they did, this is work from the 90s and 2000s. And what they did in this kind of work is that they subjected the fly to a stimulus, so visual stimulus, light, that looked like this. It was like some random noise. And then all of a sudden, they increased the magnitude of the noise, right? And what they observe is that as they, right after the increase, you see a big change in the response of here of a neuron. And then you see this, typical kind of trace that I was telling you about that goes back to some lower level, right? But what they, the interesting thing here is that there's a certain time during which you get these switches, and then there's another time, which is the time it takes to adapt. And what they did in this experiment is that they actually changed across the experiment the typical time the stimulus would stay with the given variance, right? So this time of the block here where the variance is high or low. And when they changed that time, so here, I'm not showing it, but when they switched more quickly, basically, they actually saw that the system adapted also more quickly, right? And if you actually plot this adaptation timescale as a function of the timescale with which the environment switches, they found a linear relationship. And in a way that this sort of makes sense, you would think, okay, if I expect that the environment will change slowly, then I should also adapt slowly, right? And if I expect that the environment is going to change quickly, then I should also react more quickly, right, to be able to track the environment. But is this the optimal thing to do, right? So this is the question we asked. And then to do this, we're going to use our optimality framework. But first, and so this is for, sorry, I forgot to say, this is for variance switching, but then, you can find, and this is for the fly, but you can, people found similar results for vertebrate retina and also for adaptation to the mean. Okay, so to make progress and to do our modeling, I actually need to define this objective function that I'm trying to maximize, right? I need to say, what is it that I'm trying to do, right? And in fact, it so happens that in this system, people have a good idea what the system is trying to do, and it's to maximize information transmission. So let me explain this idea. Let's imagine that the stimulus has a certain distribution, P of S, here shown in black, right? It turns out, if you want to actually, encode the stimulus into response, a good idea is actually to use a response function that looks like this, meaning that it will have a high gain exactly at the place where you're likely to see the stimulus, right? Because, for instance, if you're in this region, right, so in the region where the response saturates, the response actually doesn't contain much information, but that's good because that's the place where the stimulus is actually not likely to be seen, right? So we actually want the response function to be somewhat matched to the stimulus statistics. What this implies is that if you actually change, for instance, the variance of your stimulus, so here you switch to a smaller variance, then the optimal response should actually be to have a sharper response with a higher gain and more focused. So the dynamic range is more focused on the region where the stimulus actually is likely to happen, right? And you can actually rationalize this mathematically by saying that trying to maximize information transmission, you can actually show in simple cases that this matches directly the response function to the input distribution. And this was shown in the context of the visual system by Laughlin in the 80s, where you actually showed experimentally that you have a perfect match between the cumulative distribution of the stimulus and the response function. But I want to emphasize that this is a very general idea. This is not just for neuroscience. In fact, people apply this idea in the context of gene regulation, in the context of early flight development, to show, so viewing the transcription factor that regulates a gene as an input, the same as the stimulus, and then the output being the transcription of the gene, and showing that in this particular case of maternal gradient, maternal morphogen, bicoid regulating bicoid. In that case, there was actually also a match between the distribution of the input and the response function that actually maximizes information. Right? So this is actually good evidence that maybe that's the right objective to think about. In fact, this is another way of looking at the same thing. Like these are different experiments, but showing the same thing. If you actually take the example of that fly visual system that I was telling you about, and if you subject the fly to stimuli of different variances, so here the different curves correspond to different variances, standard deviations actually of the stimulus. You see that actually the response function, so here each curve is a different response function, is different for each, but if you renormalize them by the variance, then they all fall onto a universal curve. So this strongly suggests that adaptation actually does this information maximization, and in fact you can measure information in these experiments, and you see that following a switch, even though both the stimulus statistics is completely different and the response function is completely different, as you can see here, the information transmitted per second is actually pretty much constant. You just see maybe a small dip exactly at the time of the switch. So this is when you switch from high variance to low variance and vice versa. Right? So this is strongly, this is to motivate that we're going to use this information transmission as our objective, right? Okay, so now I'm going to introduce the model. So I start with the simplest case where we have a stimulus that has a mean, so that can switch between two values, right? And to simplify, without losing generality, we just say that these two values are -1 and +1, and they have a switching rate given by a typical timescale tau, right? So imagine that's the mean. So of course the stimulus then is drawn with the mean plus some Gaussian white noise. And this is the kind of stimulus that you get, right? So you get some stimulus, then all of a sudden you change the mean, then you change again. So you imagine going in and out of a dark room, right? And it switches with typical timescale tau. So now we're going to apply optimal Bayesian filter. So I'm not going to go into the details, but basically what we can calculate is based on all the past information, what we believe the state of the system is, right? And we call that X. So X would be +1 if we think we're in a plus state, and it will be -1 if we think we're in a minus state. And the intermediate values mean that we're not really sure, right? And it turns out that we can actually, in the system, we can derive an exact equation for the dynamics of X. Right? And it takes this kind of strange nonlinear stochastic differential equation where here Xi is the noise. And the noise comes from the fact that the stimulus is unpredictable, it has some randomness to it. Right? And this is what the trace looks like. When there's a switch to minus, we quickly adapt to a minus state. And then after a switch to plus, we quickly adapt to a plus state, and so on and so forth. Now, from this, now that the system has some belief for what the environment is, whether it's plus or minus, it can actually infer from that what is the expectation for the stimulus it should encounter, right? So if it's in plus state, it would think that it's drawn from this purple distribution. If it's a minus state, it would think it's from this orange distribution. But if it's not really sure, for instance, if it's one of these intermediate states, then it will be in this kind of intermediate, bimodal distribution, where it doesn't really know where the stimulus is going to be drawn from. Next, we apply the principle of maximizing the information transmission, and that leads to changing basically the response function as a function of time. So if you're in this purple stage, then you think you're going to be in a plus state. Therefore, you adjust your dynamic range to that mean value. And if you're in a minus state, you're going to adjust it to a lower value. right? And then when right after, during the adaptation phase here, you're going to transition from these two response functions. So the nice thing about this is that if you actually then simulate the kind of response you should get, so the optimal response, it would look exactly like the kind of thing you see in the data, right? Right after a switch, you're actually not well adapted just yet, right? So all of a sudden, you were on this you were on this orange curve, and all of a sudden the stimulus are drawn from here, so you're going to get very high response. But as time goes by, you'll adapt. By adapt, you mean that your response function is going to shift to the right, and therefore your response is going to go back down to normal value. Right? And then when you do the switch the other direction, you get the opposite behavior. Right? So you can, with this kind of very simple model, you can basically explain the kind of traces of adaptation that you see in the data. Right? So you can go a bit further, because you can actually also predict what's the adaptation timescale. So you can actually measure from the, so you can actually calculate from the model how long it takes to adapt, right? And it turns out that this depends, of course, on tau. It also depends on what we call an adaptation parameter, which is some sort of a signal-to-noise ratio, or an inverse signal-to-noise ratio, because theta is the noise. And it turns out that the answer depends on alpha. So if you're in the low noise-to-noise signal-to-noise ratio, so this is this blue curve, then it turns out that the time to adapt is actually scales almost linearly with the time of the environment, right? So the time with which the environment switches. This is what we saw in the experiment, but I have to point out that this is in the case of very poor adaptation, right? Because this is when you have a very low signal-to-noise ratio. Therefore, actually, their adaptation doesn't work. In that case, you don't really get adaptation at all, actually. In the limit where you do get good adaptation, it's when alpha is small, so you get a high signal-to-noise ratio. In that case, it turns out that this adaptation timescale is strongly sublinear in tau. In fact, it mostly depends on this other timescale, theta, and only in a logarithmic manner on the timescale tau, right? So that's interesting, because it actually means that it's not the optimal thing to do, to adapt your timescale directly to the timescale of the environment. What really drives the timescale of adaptation is how much you trust your signal, and that's given by theta, right? So there is a small influence of the timescale of the environment, but it's actually quite minor compared to what you would have expected naively. We can do the same game for variance switching. So instead of, so these calculations where we can do almost exact analytical progress is for mean switching. For variance switching, it's a bit more complicated, so we need to recourse to simulations. But we can play the same game with the model, and we can also see the same kind of trace as we did for the mean switching. And the important point is that we also see a sublinear scaling of the adaptation timescale with respect to the switching timescale. So, I mean, now that leaves the question open of why. So the experiments suggest that this is actually not the optimal thing to do. But I think this is the first interesting step to asking, what is it that the system wants to do, right? Because it could very well be that, as I said, we do see the linear scaling in a case where adaptation doesn't work. That's a case where the signal to noise, the noise is very high. Maybe that's what the system expects, and maybe it's optimized for that. Right? Because I should point out that this kind of experiments show very artificial stimuli, and the natural system doesn't see this kind of statistics necessarily. Now, we can also look at information transmission. I told you that's the thing that's optimized. And we can see, just like we did in the experiment, that it's mostly constant between one switch and the next, with a dip right at the transition. And we even see an asymmetry in the size of the dip in the case of variance switching that's also seen in the data. So that's also something nice that we can predict about the experiment. Okay, one more thing is, So, I showed you this equation for the belief dynamics, and you can see it has this nice nonlinear form. And because this is very, reminiscent of a Langevin equation in physics, so we may want to rewrite it this way. Other than dynamics under an effective potential. And so what you can see with this kind of formulation is that if you actually do, so when a system is adapted, it's actually sitting in the lower part of this potential. And when you switch y, you basically switch the shape of this potential, and then your system becomes here at the high energy state. And when you adapt, actually what happens is that your system will relax back to the other adaptive state. And that's nice because it sort of gives us an explicit analogy with the stochastic thermodynamics, and we can actually estimate how much heat was dissipated during that process by analogy with physics, right? What's also nice is that this heat that's dissipated, that's a hallmark of adaptation, that's a hallmark of the irreversible nature of the system. You can actually estimate it in an agnostic way by looking at entropy production, which is the divergence between the probability of forward trajectories versus backward trajectories. And it turns out that this entropy production is exactly what we measure with this heat dissipation. And so we can calculate that on the system, and we see again this logarithmic dependency. Right, so you can see that in the regime where you adapt well, right, which is the regime of small alpha, you actually get a large entropy production, whereas if you don't adapt, then you don't have any entropy production at all. What's nice about this measure is that it's actually, so here I can calculate it exactly in this model, but when you look at this formula, it's completely non-parametric and model-free. So in principle, it's something you can actually directly estimate from experiments. And so we propose that maybe this could be used as a universal measure of adaptation. And this is not an entirely new idea. People have also explored this in the case actually of E. coli chemotaxis, relating energy consumption and therefore this entropy production to the quality of adaptation. Okay, so now let me move on to the second application of this adaptation and Bayesian prediction, which is work from we did with Ian Emelman. So let me start with the problem we started with, which is the problem of the physical limits of concentration sensing. So let's imagine you have a small receptor of size A, and this receptor can bind some ligands, and the goal is to try to estimate the concentration of ligand just from the occupancy of this receptor, right? And so people have figured out what's the best performance you could get, and essentially the argument goes like this: is if you get n binding events, the number of binding events you should get. So if you have a measurement device of size A, it's the number of particles that can coincide with this object, which is given by the volume time to concentration, times the turnover rate, times the integration time, and it gives you this factor. And then the kind of error you should get on the estimate of the concentration you get from these binding events should scale with 1 over DACT, right, from the law of large numbers. So this was done by Berg and Persson and then refined in further work. And this is a quite relevant question, both in cases like chemotaxis, where E. coli actually has very little, tries to really estimate concentrations, very low concentrations, with only a few molecules can be around E. coli. And it's also been shown to be relevant in the case of, again, flight development and gene regulation, where in that case, it's not the measurement device is not E. coli, but instead it's a transcription factor binding site or promoter, which tries to measure the concentration of transcription factors. And there it's also been shown that the system works close to the physical limits given by this bound. So we wanted to revisit this bound in a wider context, because here the idea is that the receptor is trying to measure the concentration. It has some time t to do it, right? But in practice, the concentration may vary, right? So maybe not in flight development, but for E. coli, certainly, right? The concentration of the chemoattractant will vary over time, and it will vary with some time scale tau. Right? Now the question is, given the state of the receptor, can I try to reconstruct the the trace of C? Can I try to reconstruct the concentration? And again, I'll go I'll reuse my framework. The environments, what we're trying to know is the C of T, so the concentration as a function of time. But what we actually see is the state of occupancy of the receptor, right? Which follows also the state graph process, bound, unbound, bound, unbound, right? So it's this O of T. So now the name of the game is to basically predict as well as possible the C of T. And so in that case, our objective is just to make the smallest error as possible, right? And we can do this again using, Bayes' law. And so, we do a change of variable that's not really important to take the log of C where it's more relevant. And we also assume that log of C follows a random walk to model the fact that it's changing as a function of time, right? So with some time scale tau. And if we do this, we can show that the Bayesian problem can be reformulated as a field theory problem. So, giving this path integral, so that the exact, I'm not going to go into details of this thing, but the Bayesian theory allows you to do it dynamically and solve that thing exactly, right? So the way it goes is that when you don't see any binding events, or between binding events, you should update your belief system with this equation. And when you see a binding event, you should update it according to that. Okay, so these are the rules. But it's simpler to view them as just tracking the expected value of the belief. So that's actually our estimate of what the concentration is based on past and on the past of the receptor occupancy. And if we do a Gaussian approximation, we can actually show that then these two equations simplify. And to the following thing is that when we actually see a binding event, so here in red, then we should increment our estimate by this factor, which is given by the square root of C. And when we don't see anything, then we should actually let our concentration decay with the power 3/2, right, with this factor. Another way of looking at this is that basically when we see a binding event, we want to say, okay, yeah, I think that increases our belief that the concentration is high. And if we don't see anything, it's going to decay. But where we're going to do this with a typical timescale given by this memory timescale, right? Given by tau over for dA. And you see the square roots. Sorry. Yeah. This memory timescale is actually plays the same role as the adaptation timescale in the first part of the talk. Right. This is how much we want to remember. This is also, you can also view this as a relaxation timescale, so an adaptation timescale. And you can also view this as, this is how much of the past I want to remember. How much do I want to integrate over? And you can see that it scales again sublinearly with the timescale of the environment. Right. In this case, square roots instead of a logarithm. So now, what does it mean in terms of sensing accuracy? So if we can now estimate the typical error we make with this estimator, which is, again, it's the optimal Bayesian estimator, given the state of the receptor. And we find that it scales like 1/DAC tau, but not this time to the power 1/4, right? And this is maybe, initially somewhat surprising, because if we compare to the classical bound I was just telling you about from Berg and Purcell, we found that it was 1/DACT to the power 1/2, right? So what's the difference? But the difference is that here, my T is not my tau, right? What I need to do actually is, where I need to replace this big T by is this integration timescale is my memory timescale, right? And this memory timescale is not tau. It's instead the square root of tau divided by this rate of, basically this is the rate of arrival of particles to the receptor. So it's kind of do a geometric average between the timescale of the environment and the timescale of the events themselves. And this allows you to just reconcile the two. Okay, so I mean, this is nice. This will give you a optimal adaptation scheme. But then the question is, can any biological system actually implement this? And so we don't know whether a real biological system does implement this, but we still asked whether it was plausible. And so we could actually come up with the biochemical network that allows for this thing. And what it looks like, it really looks like an adaptive network where basically the signaling will itself depend on what you think the concentration is. So in this sense, it's really adaptation, because the response will itself depend on what the system has seen in the past. And that goes both for what happens for when you signal. So when you get binding events, you should signal a certain amount given itself by the cellular system. And also between binding events, this should also decay according to this rule. And you could do this by having a intermediate molecule B or B star that would actually be take the square root of the output. And we tested this in simulation and showed that not only it actually manages to get the right gets the concentrations correctly, but also we checked that actually reaches our new Bergen-Purcell law, this 1/4 the ACT to the power 1/4, right? Because this is the squared of the error. So here, both production and decay are regulated by C. And before finishing, I just want to mention that Peter Hein, who's going to talk tomorrow, has also done some work on a very related question. And with this, I'll conclude. So here I outlined some general principles of predictive adaptation. So one key idea is that the response should match the input statistics. And another key idea is that the memory timescale, or this adaptation timescale, depends not only on the timescale of the environment, but also on the signal-to-noise ratio. And finally, we propose this idea that this adaptation can be measured in some somewhat universal way using tools from statistical mechanics, such as entropy production. Such as entropy production. And with this, thank you, and I take questions.
Okay, thank you, Thierry, for this nice talk. So we have some minutes for questions. So I see some hands. So Ayan Biswas.
Yeah. Am I audible?
Yes.
Yes.
Thank you, Thierry. That was a very wonderful talk. So I was just wondering, so you modeled the first part of your talk using a wide Gaussian noise. Suppose the signal has some correlation, it has colored noise. So I can guess one time point within its neighborhood, immediate neighborhood, there will be some amount of common information. So does that affect the adaptation with respect to mean or variance?
I mean, thanks for the question. I think that's a great question. We actually haven't looked at this, but I think as long as so if you have colored noise, it depends on the scale of that noise. So here, in a way, we sort of assume that the time scale of the noise of the stimulus was small compared to the other time scales. If the two time scales are going to be comparable, then it's not so clear that we can view the switching thing as because then the environment is itself what goes into the correlations of the noise. You see what I mean, right? Because then you lose the Markovian nature of the stimulus. So you can always say, okay, maybe there are memory effects in my stimulus, which is what you're saying by colored noise. But then these memory effects now become part of the environment. So it depends whether you want to put them as part of the signal or part of the environment. But so that's a good question, something one can look at. I think it will make the equations a bit more complicated, but maybe still tractable.
So in general, you have your signal fluctuating in a, I know the frequency is smaller for the signal, right? With respect to the downstream stuff, whatever is adapting to it.
Right, so the adaptation is always faster. The optimal adaptation is always faster than the time it takes for the environment to change. Because otherwise adaptation would be always useless, right? So at worst, it takes half.
So can it be compared to the Nyquist theorem? So to code a signal, you have whatever is coding, so that has to fluctuate as a bigger time scale, no?
I mean, yeah, I don't know. It could very well be. I don't know, because in Inquis theorem is, I guess, I'm thinking of kind of a, Fourier modes. And here it's really highly stochastic, but there could be a connection. Yeah, thanks for the question.
Thank you.
So we have another questions by Peter Rentenwolde.
Yes, thanks, Thierry, for this wonderful talk. Let's see, I have more than one question, but let me just start with a very qualitative question. So you showed this, you discussed this irreversibility, right, so sort of the heat release during this adaptive response. And then you sort of drew this with this sort of bistable potential with a barrier there. So does this barrier come out of the model? And if so, is there sort of a function associated with this barrier? Right, sort of this tilting of this bistable potential.
Yeah, it's a great question. Actually, it's a bit strange because, okay, I tell you what, if you look at this equation, right, there's no barrier here. Like, you always go down.
Yeah.
And the barrier comes from the Ito term from the diffusion. So I'm not sure I completely understand it, but my understanding is that this is not a true barrier. This is actually overcome by diffusion, because there's also very high diffusion here. So it's, right, the height of the barrier is almost the same as the diffusion coefficient. So like, it somewhat compensates each other. So I don't think it's a real barrier.
Okay.
So the system doesn't actually get stuck. I don't know if that answers the question.
Yeah. And maybe I can ask a second question.
Maybe rapidly after that.
Very rapidly. Yeah. So so after this response, right, you get a new stimulus distribution, and this mutual information that you then compute, is that the mutual information between the output and then the stimulus? Because yes. I was wondering, if your environment switches between two states, how important is that you then perceive the actual stimulus in that given environment, given that the environment is either in one state or the other?
It is the information between the input and the output.
But is that the input, the signal in that given environment, or is it
you don't subtract the
output in either the one?
You don't subtract the mean if that's what you're asking. Yeah, it is the You don't subtract the mean if that's what you're asking.
Yeah, well, basically,
it's the input in that environment.
Is that the input that you see, right, that has much more information in there than the information, than the entropy of the environment, right, which is one bit. It's either one environment or the other.
Yeah, no, sorry. It's what you're learning is it's not the entropy of the environment. You're not trying to learn the environment. You're really trying to learn the inputs.
Yeah, exactly. So that makes perfect sense. But then indeed comes my question. Ultimately, what you care about is what you learn about your environment, right? Rather than the input, no?
Actually, here, maybe not. Here, actually, so for instance, if it's visual scene, you don't actually care about the mean luminosity. You care about the features that you see in this mean luminosity, about object.
Yeah.
So it's the information is actually carried by the contrast, not by the mean.
Yeah. No, okay, I think that answers my question. Thank you.
Thank you.
Thank you. So there are a couple of